In this video, we will be discussing certain preliminary concepts regarding the fluoroquinolone antibiotics. Now, to have an understanding about fluoroquinolone antibiotics, we need to have a rudimentary understanding about DNA topoisomerases. Now, for cells to multiply, the DNA needs to be replicated first. And the first step in DNA replication is the separation of the two strands that make up the DNA. Now, this is achieved by the enzyme helicase or helicase, whichever way you want to pronounce it. Now, this is a schematic diagram of DNA. You can see the two strands, and these two strands have to be separated, and this is done by helicase. Now, helicase does this by insinuating itself between the two strands of DNA, and you get a structure which is something like this. And as you can see, there is helicase insinuating itself between the two strands. This bubble-shaped structure that you see is what you call the replication bubble. Now, for our convenience, we shall reduce the replication bubble to what is called the replication fork, as you see here. Now, once helicase has successfully separated two strands of DNA, the next set of enzymes, namely the polymerases, can act, and they synthesize complementary strands of DNA to the parent strands in an antiparallel fashion, as you can see over here. Helicase will continue to separate the two strands of DNA, similar to how a zip is being unfastened. And as it does so, polymerases synthesize new complementary strand of DNA, and the process continues. Now, as helicase separates the two strands, we see a phenomenon happening distal to the point of separation. We see that supercoils are being formed. Now, what exactly are supercoils and what is their significance? Right. So, supercoiling means the coiling of a coil. What does that mean? This is a phone. A phone has got a cord, and the cord is in the shape of a coil. Now, as you can see, these coils can coil upon themselves, causing this phenomenon which we call supercoiling. So, as supercoils begin to form, a tension is created which tends to resist the effect of helicase as it tries to unzip or separate the DNA. If this situation is allowed to persist, then the tension created by the supercoils becomes too great for helicase to overcome, and DNA separation fails to occur. DNA fails to replicate further, and cell division stops, and at one point the cell is going to die. Fortunately, this doesn't happen because the cell has developed mechanisms to overcome this sort of a situation, and cell division will proceed smoothly. What actually happens is that under normal circumstances, supercoils are removed soon after they are formed, and this is the function of DNA topoisomerases. So, broadly speaking, there are two types of topoisomerases, namely type 1 topoisomerase and type 2 topoisomerase. Now, type 1 topoisomerase, irrespective of whether we are talking about human or bacterial topoisomerase, type 1 topoisomerase creates a single-stranded break in the double helix of the supercoiled DNA. It then passes an intact strand through the break. And thirdly, it reseals or religates the break. The supercoil is thus removed, the tension is relieved, helicase can continue to separate the DNA, and polymerases can continue to synthesize complementary strands of DNA. Similarly, if we talk about type 2 topoisomerase, irrespective of whether this is in bacteria or in humans, type 2 topoisomerases create two cuts in the DNA. It creates double-stranded breaks in the double helix of the supercoil DNA. It then proceeds to pass a different part of the intact helix through the break, and then it reseals or religates the double-stranded break, and therefore relieves the supercoil. Now, when we talk about bacteria encode two varieties of type 2 topoisomerase. They include gyrase and topoisomerase 4. Now, DNA gyrase is the only enzyme which is known to underwind or negatively supercoil DNA. Now, this is the enzyme that helps in initiating DNA separation and elongation of DNA. Now, let us look at the function of topoisomerase 4. To understand the function of topoisomerase 4, let us look at this diagram over here. This is a bacterial cell with its genetic material. This cell needs to divide into two daughter cells. For this to happen, the genetic material needs to replicate. Let us now replicate the genetic material of this bacterial cell. It is in the form of circular DNA in this example. So here we have successfully replicated the genetic material. And ultimately this parent bacterial cell becomes two daughter cells, each with its separate set of genetic material. However, there is an intermediate stage when the cell is in a state where it has completely replicated its genetic material. However, the replicated genetic material is in a state of catenation. The two sets of genetic material are catenated. They are linked together like the links of a chain. This is what we call catenated DNA. For this catenated DNA to separate from each other, double-stranded breaks need to be made. The two sets need to be separated, and the transient cuts that were made in the DNA need to be resealed. So, topoisomerase 4 is a type 2 topoisomerase. It creates double-stranded breaks in the DNA. And it's very, very significant when we need to decatenate catenated DNA. Right, now we can move on to the mechanism of action of fluoroquinolones in detail. So, fluoroquinolones inhibit type 2 topoisomerases in bacteria. It inhibits both DNA gyrase and topoisomerase 4. By inhibiting DNA gyrase, more and more supercoils keep forming. There is increased tension to the activity of helicase. At one point, helicase will fail to unzip or separate the two strands of DNA further. DNA polymerases fail to replicate the DNA, and this cell is going to die. So the inhibition of DNA gyrase activity by fluoroquinolones is more significant in gram-negative bacteria. Fluoroquinolones also inhibit topoisomerase 4, in which case catenated bacterial DNA remain catenated. They fail to separate, and in such a case the bacteria fails to progress further beyond the stage of division and ultimately undergoes cell death. So this is, in a nutshell, the mechanism of action of fluoroquinolones. Right. Now we come to certain important features of fluoroquinolones. Before fluoroquinolones were introduced, there were only quinolones, and the first quinolone was nalidixic acid. When this structure was fluorinated, we came up with fluoroquinolones which were effective orally and which had broader antimicrobial activity. So fluoroquinolones may be classified into multiple generations depending on your source from which you quote the generations will be different. So the first generation fluoroquinolones were introduced in the 1980s. They included ciprofloxacin, ofloxacin, pfloxacin, and norfloxacin. Ciprofloxacin is the prototypical drug in this group. Pfloxacin is safe in renal failure, and norfloxacin is the fluoroquinolone with the lowest bioavailability. Right. The second generation fluoroquinolones retain their effect on gram-negative bacteria, but they have additional effect on gram-positive bacteria as well. The second generation fluoroquinolones include moxifloxacin, gemifloxacin, sparfloxacin, levofloxacin, lumifloxacin, and prulifloxacin. Now, certain important points to remember regarding these drugs include the following. Moxifloxacin is the fluoroquinolone with the second longest half-life. Sparfloxacin has got maximum photosensitivity. It has got the longest half-life and produces QT prolongation. Now, regarding levofloxacin, it is the fluoroquinolone with the highest oral bioavailability. And regarding lumifloxacin, we need to remember that it produces photosensitivity as well as QT prolongation. Now, certain fluoroquinolones are called respiratory fluoroquinolones simply because they are very effective in respiratory tract infections. They include moxifloxacin, levofloxacin, and gemifloxacin. Traditionally, the teaching was that fluoroquinolones are not at all effective in MRSA infections, and that there are only two fluoroquinolones which are effective against Pseudomonas aeruginosa. These two fluoroquinolones include ciprofloxacin and levofloxacin. However, there is a new fluoroquinolone that has been recently introduced that seems to have decent activity against MRSA and Pseudomonas aeruginosa, and the name of that fluoroquinolone is dilafloxacin. Now, fluoroquinolones do have their adverse effects, and these include QT prolongation, tendinitis, and tendon rupture. This is a rare but serious adverse effect of fluoroquinolones. Other important adverse effects include Clostridium difficile associated diarrhea, as well as CNS manifestations like hallucinations, confusion, and so on. Rash, anaphylaxis, and aortic dissections and aortic aneurysms as well. These are also adverse effects associated with fluoroquinolones, rare but serious. Fluoroquinolones are also known to cause photosensitivity and hypoglycemia. A mnemonic to remember these adverse effects would be QT crap. The uses of fluoroquinolones are very wide. They may be used in prostatitis, in UTIs, urinary tract infections, in bone and joint infections, in gastrointestinal infections, in sexually transmitted diseases, and in respiratory tract infections as well. The mnemonic for this would be PUBG sir. Now, there are topoisomerase inhibitors without antimicrobial activity but with other pharmacological properties, and it is useful for us to study these as well. So topoisomerase inhibitors without antimicrobial activity but with anti-cancer activity may be classified into two types. Type 1 topoisomerase inhibitors, but in humans, and type 2 topoisomerase inhibitors in humans. Right. So, the type 1 topoisomerase inhibitors in humans are called the camptothecins, the examples of which include topotecan and irinotecan. The type 2 topoisomerase inhibitors in humans include two classes, namely the anthracyclines and the epipodophylotoxins. Examples for the anthracyclines include hydroxydaunorubicin. Now, this is a drug which is known by three names. The first name is hydroxydaunorubicin. It is also called doxorubicin. It is also called adriamycin. So, one drug with three different names. There are other examples in this group. They include idarubicin, daunorubicin, epirubicin, and mitoxandrone. Right. The examples for the epipodophylotoxins include etoposide and tinoposide. Right. All topoisomerase inhibitors that act as anti-cancer drugs inhibit the religation process. They inhibit the resealing process, and therefore this ends up in fragmented DNA in these cells. The rapidly dividing cells in cancer fail to replicate further. So this forms the basis of the mechanism of action of topoisomerase inhibitors as anti-cancer drugs. Right. So, very briefly, type 2 topoisomerase inhibitors for bacteria include the fluoroquinolone antibiotics. They can inhibit DNA gyrase and topoisomerase 4. Now, type 2 topoisomerases may also be inhibited in humans. There are no antimicrobial drugs, obviously, for this indication, but we do have anti-cancer drugs for this indication. They include the anti-cancer agents such as anthracyclines as well as the epipodophylotoxins. Interestingly, the type 2 topoisomerases in humans do not include gyrase or topoisomerase 4. Instead, they include topoisomerase 2 alpha and topoisomerase 2 beta. Now, there are also type 1 topoisomerase inhibitors in humans. They include the anti-cancer agents such as the camptothecins.